principal objective of the European project, Life 13 Evergreen, is to reduce land pollution from traditional pesticides by substituting their usage with that of bioactive molecules derived from vegetable compost, in particular by using polyphenol products extracted from non-food-based biomass waste. The identification, experimentation and validation of these molecules represents a viable alternative to both current commercial pesticides and also to copper salts, both in conventional and organic cultivation techniques. The project participants are DISPA, D-I-S-P-A-A, of Florence, INSTM, CBAS CSIC of Murcia, Astra SRL and Mondo Verde SRL. The project leader was DISPA of Florence and the coordinator was Professor Stefania Tegli.
Evergreen began where after CU ended. The Evergreen project was all about the study of plant pathogen bacteria and nematodes. We were looking to find alternatives to the use of copper salts and traditional nematicides that were capable of combating plant diseases, limiting the onset of disease and any eventual symptomatic spreading. All of this without impacting negatively upon the environment, human health and so on. We were looking for molecules that were not antimicrobic but anti-infectious, because it is this that makes the difference. In short, not to overly disturb these living populations of bacterial phytopathogens, but simply to disarm them. We looked for these combative bioactive molecules against bacterial phytopathogens and nematodes within the grand family of phenols. So, what was our thinking? We extracted polyphenols from these plants and we used these prototype preparations, constant in both their qualitative and quantitative properties, and then administered them to plants in order to determine whether or not we could protect them from these pathogens. An important element of the project was that these polyphenols were extracted from parts of the plant destined to become waste.
The Evergreen project was developed in 3 integrated phases. Firstly, the choice of bacterial models, nematodes and plant models on which to base the trial. Secondly, the verification, initially in the laboratory, then on a pilot scale, of the performance of polyphenol extracts obtained from the waste of olives, artichokes, grapes and chestnuts, all obtained by eco-sustainable methods. Finally, having obtained a sufficient quantity of useful polyphenols, we then moved to field trials.
DISPA's role in the project was to verify in the laboratory the eventual ecotoxicity of the polyphenolic composts and their efficiency as anti-infectives against phytopathogenic bacteria.
My name is Professor Biricolti and I work at the Department of Agricultural Science, Food and Environment. I collaborated with Professor Tegli and I'm responsible for this laboratory. The scope of this project was to study the interaction between pathogens and plants in controlled conditions. We grew the plants in vitro, i.e. in conditions which gave us the opportunity to control many of the environmental, climatic and biological parameters. This gave us the ability to get right to the heart of the effects of the polyphenolic molecules, with the main objective of the project, and in this way to observe how these influence the interaction between the pathogens and the plants. The results have been most satisfactory regarding the application of these polyphenolic molecules, which derive from a variety of natural origins. In particular, some of these have given us very interesting results, offering precise indicators to the project collaborators.
En este proyecto, el trabajo que hace el grupo de investigación del CEBAS.
In this project, the work carried out by the research group CBAS is principally concerned with the soil and those issues that can emerge when one introduces any element that is not normally present. In this case, the polyphenols.
Caso, como ahora explicaré, dentro del proyecto Evergreen.
This project studies what happens to the soil structure when one introduces a foreign body.
Ese elemento extraño se introduce dentro del suelo. Nosotros, en este proyecto, como digo, el grupo del CBAS-SIC, que tiene un experto.
Given that Evergreen is fundamentally an environmental project, we have entered the world of polyphenols. We have concentrated on those polyphenols derived from chestnut and grape waste.
Introducir polifenoles derivados fundamentalmente de castaño y de residuos de la uva, de la vid. En el trabajo que nosotros hemos llevado a cabo, inicialmente lo que hemos
In the beginning, we verified whether or not these polyphenol extracts from vegetable waste could be used without difficulty as biocontrol agents or as pesticides against plant pathogens and nematodes, because they don't overly disrupt the biodiversity of the actual soil.
Porque no inciden negativamente sobre la biodiversidad de ese suelo. Nosotros también, dentro del grupo de trabajo, hemos
We
llevado a
experimented upon two types of plant, the kiwi and the olive, and also with the tobacco plant, which is easily attacked by nematodes. We investigated if the action of the polyphenols from the Evergreen project effectively combated the nematodes and if they had any impact, positive or negative, on the soil.
incidencia positiva o negativa sobre el suelo. Desde luego,
I emphasize, as regards the soil, no problem emerged, and that in biological competition terms, polyphenols can be considered effective in the control of bacteria and nematodes.
claro, frente a ese tipo de bacterias y a ese tipo de nematodos.
Astra, comunità operativa come partner del progetto.
Astra, one of the commercial partners in the Evergreen project, has closely followed the experiments from the beginning, both the pilot and the field testing, for the effectiveness of tannins and polyphenols in the control of bacterial diseases. Taking into account the situation in general and the types of cultivation present in the region of Emilia-Romagna, we have been primarily researching the olive and the kiwi. Furthermore, we have verified the efficiency of tannins on the nematodes, on seed-bearing beet and on carrots.
Bietola portaseme e quest'anno su carota. Abbiamo verificato cultura.
We have used potted crops, thereby controlling precisely bacterial diseases introduced artificially.
Nel caso del kiwi abbiamo ottenuto
In the case of the kiwi, we have carried out artificial inoculations and we have verified the effectiveness of tannins in the control of these bacteria. Also, in the case of the olive, we carried out artificial inoculation in trays in greenhouses, and then we verified, through a series of assessments, the effectiveness of the tannins upon the plants.
valutazioni della pianta quello che era l'efficacia dei tannini. Dalla fase di serra siamo passati alla fase di pieno campo.
From the greenhouse stage, we passed on to the field stage, both here and in other locations, where we verified the usage of these products in actual agricultural production.
Il progetto Evergreen è stato sicuramente per noi qualcosa molto
Evergreen is most definitely a project that is close to our own DNA. Our specific function in Evergreen has been that of offering technical support in the derived formulations coming out of the university research. We had to create the practical formulations and contribute to the subsequent industrialization and commercialization of the extracts.
la classica informazione.
The information that emerged from the work of Professors Tegli and Romani have been fundamental to all the project partners. It should also be added that the information and ideas exchange has been truly universal between all partners, and this has been most gratifying.
Tutti i partner ed è stato anche veramente soddisfacente. Sicuramente questo progetto ha una...
Without doubt, this project has huge potential. For now, let's call it theoretical. exceptional. To use natural products almost entirely toxin-free is something that is most compelling and will no doubt be enthusiastically received by all operators in the sector.
The work of DISPA, as I have said, has been both that of a coordinating role and that of direct and demonstrative research, and that has been conducted not only by the teaching staff, of course, but as always, thanks to the young, skilled and dedicated researchers, for the most part on a non-permanent contract.